Hello everyone and welcome to Life Science Prep. Here I'll discuss some topic on CSIR NET and try to summarize all the things about cell signaling, other topics too, but for now cell signaling. So what is cell signaling? This is communication between the cell and their environment or a particularly source cells or to the target cell. Here, the source cells release some signal molecules from a particular distance or nearest to it, such as chemical messenger, hormone, neurotransmitters, etc. Anything that has been received through the target cell through a particular receptor that sends or detect chemical signals or chemical messengers and transit it to the target cells, and the target cells responds to the signals. So the cell signaling divided into two types, that is direct cell signaling or indirect cell signaling. Direct cell signaling is a type of physical contact between the two cells. Through physical contact, the signaling has been transferred from one cell to another. But in indirect cell, there are three ways through that the cell transport signal to the targeted cell. So the direct cell signaling, that is cell to cell contact here, mostly the two cells come closer at close proximity where they can directly contact or share the signal through their plasma membranes or some particular receptors or some channels. Uh, here in animals, such gap junctions are present, that is formed by connection proteins where the signals occur, and in plant, plasmodesmata is the great example through that uh, some aqueous chemicals or some different types of mm, ions that has been transmit from one cell to another cell. Indirect cell signaling. Here, mm, the signaling is divided as per distance. So for autocrine, that is for self, paracrine, that is nearest or close proximity cells, and endocrine, that is distance distant cell. That is we can say that from hypothalamus to adrenal gland or kidney, etc. We can map that as, for example, uh, let's see the as for graphical structure. Here, autocrine signaling, a particular cell release the hormone or signal molecules that has been received by itself through its receptor. That is autocrine signaling. But in paracrine signaling, here the close proximity cells, that is uh, nearest to it, has been released a signal that received through the target cell, that is called paracrine. Here in paracrine signaling, the signal have less power. So if it goes to most distance or more distance, it the signal degrade. But in endocrine signaling, is transferred through blood to larger distance from head to tail, where the signal uh, did not goes to end or did not degrade. But at target cell, a particular receptor is present that will be received. No other cells can receive that. Signal molecules are of two types, that is polar and non-polar. In that polar ligand, that is hydrophilic. They are uh, they are water-loving signals. Due to their water-loving characteristic, the polar ligand can't uh, can transfer from outer environment to inner environment, passing through the plasma membrane. They can't pass the plasma membrane due to the plasma membranes have a lipid structure that is hydrophobic. So the hydrophilic polar ligand can't transfer that one. As per example, you can see that uh, in cell signal in cell the receptor does have a receptor that is outside or in the plasma membrane, intracellular receptor that receive the polar ligand, and due to they uh, can't cross the plasma membrane, they have to react there or transfer signal to the cell. But in nonpolar ligand, the ligand can transfer through the plasma membrane or cross the plasma membrane due to the plasma membrane have a hydrophobic environment. So the nonpolar hydrophobic easily cross the plasma membrane. There will be no problem. So the ligand can cross easily the plasma membrane, and the receptor always present for nonpolar ligand is inside the cell. So it's directly bind in the cytoplasm or in the nucleus, anywhere inside the cell. So it can easily be cross the plasma membrane. So there will be no problem in their receptor. There no need to present in the in the cell, in the plasma membrane, intracellular layer. There will be no problem for them. As per today, we discuss so much. You should read the particular part, and for next topic we'll discuss later. Thank you.